Hi, my name's Dr Adam Booth and this is another locked down edition of Quantum Source. Thanks very much for tuning in. Now, without getting too serious for a minute, this might be one of the more important quantum sources that we've ever delivered. Obviously, at the moment, we're in lockdown conditions as a result of the coronavirus. One of the ideas that I had when putting together Quantum Source was that if we could bring real life researchers to you as the general public and you could hear them talk about the science that they do, the methods that they use, and the kind of data that produces, that it would equip you with a little bit of a better bullshit detector, so that when you're out there looking at social media or listening to fake news or whatever, you can make a call for yourselves about whether someone is actually representing scientific data in the way that they should be doing. And of course that's really important in the current situation, where there's decisions being made on a day-to-day basis related to viruses and virology. So today's speaker is Dr Cheryl Walter, and we're going to give her a call in just a second. She is a lecturer in microbiology at the University of Hull, where she works on viruses and virology. So we'll be giving her a bell. Settle in with something nice. My recommended source for this particular quantum source, I'm going to pass over to Beth at the Constitutional Club. She's going to give you a cocktail recommendation, and it features a particular kind of Mexican beer that you usually serve with a lime in. So I'm going to pass over to Beth. You can download this at the Constitutional's Facebook group.
So today we're going to be making a michelada cocktail.
Ingredients wise, I've got some fresh lime here, some hot sauce, any hot sauce will do, Tabasco, whatever you've got in your cupboard, some crushed chilies, some sea salt, and last but not least, a lager of your choice. Now with the lager, the lighter the lager, the better. So if you want to use a Modelo or a Corona, if you'll pardon me for saying Corona right now, that would be the best.
So first thing you're going to do is you're going to chop up two.
And you can watch the rest via the Constitutional's pages, but I'm going to give Cheryl a call now. And hello, Cheryl, there you are. How are you doing?
I'm fine, thanks, Adam. How are you doing? It's nice to chat to you today.
Absolutely, yeah. I'm coming to the end of a long week actually, so I'm looking forward to the weekend, and yeah, it's going to be a good one. Locked down, but it's not work. So.
I'm glad you can tell the difference between a weekday and a weekend. I've long since lost that skill.
I've kind of definitely lost track of the number of days that we've been in this situation.
Oh yes, I agree.
So, with all this lockdown tedium, give me a sauce that's keeping you sane.
Oh, good old Worcestershire sauce.
All right.
And you can add that to pretty much any cooking, any savoury cooking. One of my favourites and go-tos, and I chose it for a reason today. It is very versatile, very universal, pretty much like viruses. So that's my sauce.
Right. That's the kind of tenuous link that we like at Quantum Source. On that note, viruses and virology. We're obviously all affected by viruses at the moment. And can you give us the lowdown on it?
Well, I cannot believe the situation. I've been lecturing on epidemics, pandemics, and the so-called disease X for a number of years, and I never thought in my lifetime we would have a situation like we have right now.
Right. It's like living your practical research in real time.
Absolutely. We knew we were overdue a pandemic. It sounds pretty faithless saying that, but we were overdue a pandemic. Scientists predicted it was going to be flu. But in the space of 4 months, we have obviously a pandemic coronavirus that is spread all over the world. And it's unusual for a variety of reasons. And it is challenging too. It's incredibly challenging. It's very infectious. And obviously has high mortality rates. It has variable symptoms in some people, so you might be contagious and not know it. So it has just been a plethora of unusual circumstances, really. That's probably why it's one of the reasons why it's ballooned to the extent it has.
I'm kind of like weirdly reassured that you find it, you as a virologist find this just as mind-blowing as I do as a layperson when it comes to virology. The rapidness of the spread.
Absolutely, I can't believe how quickly it spread. How long had it been around for? We don't know.
Mm.
Perhaps it had been circulating in a population for a while and we didn't realize. Perhaps there was more than one spot in China, which is where it's likely to have originated. And those spots, those kind of focus points, have kind of grown at the same point. There are so many unanswered questions.
Mm. On that note, let's get some questions answered then.
Let's do so.
If I could get you to share your screen.
No problem. Right, so I titled my slides today, Viruses Neither Fish Nor Fowl, and I'll get back to that title. And I've only got a few slides, and I thought we could talk around them, and it's not overly structured or anything. So please fire ahead at any point, Adam, if you've got any questions.
Will do.
But I've taken these slides from some of the outreach that I do, some of my lectures, and I've tried to make it interesting. And I think we've been inundated, haven't we, with virology, like we're probably never going to be ever again, hopefully not. So we've all learned a little bit about viruses and virology over the last 4 months. And hopefully this tops up what everyone knows and maybe answers some questions that people have. And maybe it sets a few new questions in your mind too, things that you might be interested in. So what are viruses and why do we care? Well, firstly, I hope by now you can tell the difference between viruses and bacteria. Very, very different, both infectious, different sizes. Viruses are pretty much tiny little parasites, whereas bacteria can plod along on their own a lot of the time. Mm. And bacteria have their own metabolism, just like we do. They can tick on by. They can evolve. They can replicate. They can change. And viruses can do some of these things, but not all of them. But the main difference between the two is that viruses are a lot smaller, tiny in comparison. Mm. And of course, I hope you all know, if you've got a cold or a flu, antibiotics do diddly squat for viruses, and you've got to take something else. And think about this question. It's probably a little bit edgy, but anyone listening to this webinar, and you included, Adam, do you think you have a viral infection of any sorts right now?
I certainly don't feel like I've got a viral infection of any sort, but I'm guessing you're going to tell me that we all live with viruses, whether we even know it or not.
We do. There are lots of different ways of answering that question, but some of the tools that we've got at our disposal and some of the studies that have come out of it have suggested that at any one time, you and I have got 6 viruses in our bodies circulating. And obviously, most of the time, they do nothing. If you've ever been infected with chickenpox when you're a child, you never get rid of that chickenpox. It stays in your body, and it's mostly just completely silent. And for the most part, those viruses that we carry around with us do absolutely nothing. Sometimes you obviously get an acute infection like a cold or a flu, and they infect you, and then your immune system mounts a response and they go away. But viruses make up part of us, and you've heard of the term microbiome, and we probably have a virome too. And that virome might be important. It might be a two-way street, or it might have a clinical significance. So most of the viruses we come across, and this new coronavirus is probably one of these, is acute, and it's self-limiting. It doesn't stick around forever, but many are persistent. Many are chronic. Examples like HIV. If you get HIV infection, it's a persistent chronic infection for the rest of your life. And it's the same for Hep B and Hep C. So as scary as those 3 sound, we've actually got very good drugs which control all 3 of those. But there are some viruses out there that are chronic, that our immune systems cannot clear. So kind of carrying on from that thought, as we are all acutely aware, there are more and more emerging viral threats out there. And viruses are emerging and popping out of the woodworks for a variety of reasons. But one of the biggest reasons is population growth, human population growth. Our numbers are absolutely huge compared to what they were in the mid-1800s. I think the population in the mid-1800s, just before the Industrial Revolution, was about 250 million people. So we travel more, we need more to eat, we need more space to live. We are farming more, and the climate's probably changing because of all these things. And all of those factors contribute towards why we are seeing the steady increase of these weird and wonderful viruses popping out there.
A devil's advocate question there, in that when we get better at recognizing viruses and sort of measuring them, does that just mean that we measure more viruses, and so it's something of like an artificial effect?
Absolutely. I think, you know, when you go fishing, you are going to find something, aren't you? Absolutely. So I think there's a bit of that. Absolutely. And we are trying to predict and trying to get ahead of what some of these threats might be. But like you say, we now have these wonderful, powerful tools which allow us to go and look in deepest, darkest caves somewhere in, I don't know, Africa, and be able to take samples and say, wow, look at that. There's a whole population of viruses in this pool of water, for example. And some of it really is that these viruses are increasing to emerge. And that means they've popped out. They're a new virus, completely uncharacterized. So there's that. There's the exotic side of things, these emerging viruses. But also there's the more day-to-day thing. And that is, we reckon that viruses probably cause 1 in 6 cancers, which is pretty astounding. And that's a conservative estimate. Think about HPV and hepatitis B viruses, which is what that grainy stuff in the bottom right-hand corner of the screen is. That is hepatitis B virus. Doesn't look like much. Probably looks like ground glass under a magnifying glass. Hep B is the second biggest carcinogen in the world after nicotine. Wow. So that means it's the second biggest cause of cancer in the world after nicotine and smoking.
You just don't make that link. You know, you think viruses are like, you know, a cold or chickenpox or something. You just don't make that link to cancer.
They've got that a profound acute effect on our health, and they've also got long-term consequences, sometimes directly and sometimes indirectly too. And on the left here, this is just a little graph. You might not be able to read the scale, but you probably recognize trends. And these are new COVID-19 cases as of today. And the UK is outlined in blue there. And you could see, I think, the number of total cases is sitting at about 3.3 million cases today. Absolutely amazing. So now is a good time to be a virologist or be interested in microbiology. They are very important for these global acute emergencies and also those long-term consequences.
Yeah, totally.
So I ask my students all the time, how big are viruses? I used to have a giant bag of oranges. I used to pass the oranges out in class and give everyone a Sharpie and say, that orange is a cell in your body. Draw on the orange a virus particle. Give me an idea of the scale difference between a virus and a regular cell in your body. And most of them would draw quite, you know, a 50p, you know, size Sharpie circle on the oranges. And in actual fact, viruses are absolutely tiny. They're normally in the nanomolar range. And if you look at the scale, an atom is 10 to the minus 10 meters, absolutely tiny, one of the lowest things on that scale. DNA is in the nanoscale, and viruses are just the next notch up from that. So viruses are absolutely tiny. And if you did have to make that comparison of, well, give me an idea compared to a cell in my body, well, if you had to take a cheek cell from the inside of your mouth, the smallest virus is about 3,500 times smaller than that. And the very biggest, clunkiest virus we've ever found is still about 800 times smaller than that.
Wow. So
absolutely minute. It makes them difficult and challenging to work with, as you can imagine. And it's only been in the last 100 years, really, that microbiologists have been able to tell the difference between bacteria and viruses, purely because they were so small and it was so difficult to distinguish them.
Yeah. It does make me think, like, when you get people, obviously, like you think about PPE and people wearing face masks, that there's no way that the virus can't pass through this face mask. But is that because the face mask is supposed to filter the stuff that the virus is on rather than the virus itself? Okay.
Absolutely. So when we talk or we we cough, and obviously if we sneeze, you release these little aerosols, and those viruses are in those aerosols, and they're quite big. They're in the micron range. So those filters, and even quite a lot of the cloth face masks that you see people have been making at home, many of those can actually filter out those big droplets.
Okay.
The ones that you buy, the N95s that you hear about all the time, they are really, really good. They don't clog so quickly, and they can be used for a long period of time, and they can filter out those particles. And fabric ones can work to an extent to some degree.
Okay.
So the reason why I called this presentation, or this kind of chat today, Neither Fish Nor Fowl, is I often get asked, are viruses dead or alive? A true biologist would say they are dead. And the reason being is that they cannot replicate on their own. They can't reproduce on their own. But being a virologist and them being one of my pet subjects, I like to think of them as being alive. And it's debatable. They might not be able to replicate on their own. They need to infect a host to carry on that process and make loads and loads of viruses. But viruses can evolve. They can change. They can adapt. They can shrink their DNA. Sometimes they've got RNA for a genome, and they can expand it. So that very feature of them I find fascinating, which is why they kind of straddle that huge biological divide between being something inert and something alive. So I've just got a couple of pictures up here, just showing the range of different viruses out here. This big thing in the middle, which looks like it's got two eyes, this is a marine virus, and that's one of the biggest viruses known. That's a huge, big, clunky one I mentioned earlier. And Adam, do you recognize this thing that looks like a little Sputnik?
Well, it's almost like the classic drawing of a virus. You know, it was kind of like a spidery thing, but I've got no idea which virus it is. I'm not acquainted with that one, I don't think.
This type, and they all look the same. They all look like some little moon module.
Yeah.
These are called bacteriophages, and anything that looks like this infects just bacteria.
Okay.
So they're obviously quite intricate. There's lots of different structures in there. Some of them are pretty boring, like that first panel on the top left. And some of them are really interesting. You might recognize the picture in the bottom left. It looks like it's got a skittle in it. That is HIV.
Okay.
And the ones in the bottom right that look like little bullets, that's rabies virus. So they come in all shapes and sizes. You might recognize that one. The shepherd's crook is Ebola virus.
Okay.
And my dad joke for the day is this one in the bottom right that just come up that looks like a fried egg. That is chickenpox. So
Hey.
hey, someone appreciated it finally. I always get
fried eggs with Worcester sauce. It's all the virus food links.
I always get blank faces when I pull that one. So I get a lot of questions from the public about how do you measure them if they're this small and this tiny and they're inert when they're not inside a host. How do you measure them? Well, the obvious thing is you can look at them under a microscope. And microscopy has evolved hugely in the last 30, 40 years, maybe 20 years ago. If you managed to purify some virus and looked at it under a very powerful electron microscope, you just see these little spots like you can see in the bottom left-hand picture. And that's pretty inert. That doesn't really tell you what's happening when they're inside a cell, or perhaps the intermediate little particles that they make. Whereas if you look at that picture next to it, I know it's still grey, I know it's still grayscale and looks pretty boring, but it's actually a profoundly interesting picture from a virologist perspective. This is a cell. This thing on the top left is the nucleus of the cell. That's where our own DNA is kept. This little line here is the membrane, so that's what's enclosing the cell. And these little spiky things inside, that's influenza virus. Wow. So we've now moved on massively so that, sure, we don't just take these inert stills anymore. We can have a look and see, what do these viruses look like inside a cell? How are they changing? Where did they go? It's a far more dynamic process. We can also see how infectious they are. And this doesn't look like much, these purple wells, but once upon a time, the bottom of these wells were living cells. And we have diluted some virus and put them on those cells, and the virus has notched out little holes in that layer of cells. Now, we've since gone and stained the cells purple. And that is an incredibly simple, basic way of answering the question, how infectious is a virus? If I dilute it out 10-fold, 100-fold, a million-fold, do I still get these little notches? They're called plaques. Do I still get plaques when I add it to a cell line you can infect? So these are ways of visualizing viruses. You can sequence the genomes. You can sequence us, as we all know. You can sequence genes, and you can sequence the genomes of viruses, and that's very powerful for identifying what do their genomes look like. Already we have the genome of the new coronavirus, and it's huge, by the way.
Nice.
And we can also get an idea on how quickly does a virus mutate or change, which is very important if you are thinking of developing a vaccine or treating someone with HIV, for example. And slightly more techie than that, you can even look at the proteins that these little viruses make, those little shells, and you can say, right, how many of them do they make? What do they start off at? Are there different intermediates along the way? And that's, again, quite important for figuring out how do they make those little shells? How do you stop that process? Is it a 2-step process? Is it a 100-step process? So it gives you an idea on, it's pretty basic virology, but it's important for figuring out how they work, really.
Mm.
Indirectly, and this is something which everyone's been talking about in the news, is fine. Maybe 10% of the population has had an infection, any kind of infection, common cold. They have recovered months ago. How do you know they've had that infection? And an indirect method of measuring and studying viruses is to look and see if those people have made antibodies towards that virus. So as soon as we, and all animals do this, as soon as we're exposed to something foreign, you make an antibody towards it. And obviously a virus or bacterium is something foreign, and you make antibodies, and you make quite a lot of these antibodies. So an indirect way of studying viruses is to not look for the virus in cells, but it's to look for the antibodies. And so I've just got a little picture of a very basic test kit for HIV that you can buy. And you can use the same that you can, that obviously detects antibodies that people have made, but you can purify these little antibodies and use them in a clinical sense. And here you've got some cells that have lit up green because the antibody is detecting a corresponding viral protein. In this case, again, it's an HIV protein.
Mm.
So lots of different tools.
Is that the start of a treatment as well, then, or?
Absolutely, absolutely.
Okay.
In the situation we find ourselves now, anything is we're looking at absolutely everything as a treatment option. But you might have heard of in the news, there are lots of clinical trials going on at the moment for convalescent plasma from people who have recovered from COVID-19. And what's in that plasma is not red blood cells. It's not like a blood transfusion that needs typing. But what's in that plasma from those people that have recently recovered is a lot of those antibodies. And you can infuse them into someone, and they will be passively protected. They'll just automatically get a lovely donation of antibodies that will help them recover quicker. Good question, Adam. Yes, absolutely. You can use those antibodies for clinical purposes too.
Cool.
So how do you get a viral infection? And this is something which was interesting about coronavirus that is currently circulating. Airborne is the obvious one. Bodily fluids like Ebola virus. Ebola virus is often mistakenly thought to be airborne transmitted, and it's not actually. You've got to be very close to someone and get bodily fluids from them. Fecal oral route like polio or hepatitis A virus, where you've eaten some unwashed fruit or something like that, and you can pick up infections like that. Fomite transmission, and fomite, it's such an awkward word, but really all it means is a contaminated object, a shopping bag or a handle on a bus or something like that. And you've touched it, you've picked up something, and then you've not washed your hands and eaten your lunch. That's fomite transmission.
I was actually going to bring in, like, antibacterial wipes there,
Yeah.
then I think, well, we're dealing with viruses and not bacteria, so I didn't want to make myself sound stupid, but yeah.
That's still pretty good.
Okay.
Absolutely. At the moment, and with a lot of viral infections, those antibacterial wipes will work against viruses. Not all,
Okay.
but many.
Right. Okay.
So that's quite useful to know.
Sure. Yeah.
And you can get viruses by insect bites. Think of Rift Valley fever. You might have heard of Lassa fever virus. There are a couple of exotic ones out there. Dengue. And mother to child. Sometimes, in case quite unusually, but you can still get it. HIV. Some other viral infections can pass from mother to child, although it's actually, thankfully, not that common. So another big question that comes up is how stable are viruses outside of your body? Say you go and splutter on a bus handle on your way to work. How long does it take before that bus handle doesn't have anything infectious on it anymore? And that all depends on the virus and how that virus is built up. And one of the big deciding factors is, does the virus have a fatty lipid layer around it? Some viruses have them, some viruses don't. So that's quite an easy way of splitting them up. If they've got that fatty layer around them, normally they are quite labile, meaning they break down really quickly. And it doesn't take long for them to literally shrivel up and dry out and stop being infectious. With this coronavirus, it's interesting in that it does have that fatty lipid layer, but it still sticks around for a couple of days on surfaces. So that has been a challenge, and that was a surprise. Whereas some things, I think Hep B, for example, lasts for a few hours on surfaces, and then it's no longer infectious.
And so I don't want to put you on the spot, but might that suggest then that the coronavirus has been so infectious through this fomite route, or and not through an airborne route, or what?
It's a really good question, and I've read some literature on this, especially from epidemiologists who study, how does a virus do the rounds? How infectious is something? How is it being transmitted? And they reckon that the cases of fomite transmission for this particular coronavirus is actually pretty slim. They're not that common. You've got to be quite unlucky. Say you've ordered something online and the delivery man picked it up from a warehouse where someone spluttered on your box. He picks it up, puts it in his van, he drops it off the next day, he leaves it outside your door for a few hours, you pick it up. The chances of you picking up that box right on the spot where someone spluttered on it, still getting a decent infectious dose after all that time and still managing to somehow put it near your nose or your face or your mouth is actually quite slim. So thankfully, it doesn't happen very often, especially in this current situation.
Okay.
So some good news. So a thing that comes up is, well, how do you kill a virus? And everyone, I don't know if you can see this, everyone's probably got these little bottles of hand wash. And they do work in the current situation. They dry out that fatty lipid layer that this coronavirus has. But you'll see in the writing on these little bottles all say that they're antibacterial and they kill a certain percentage of bacteria. They don't kill all bacteria. And they don't kill all viruses. And remember that if a virus doesn't have that fatty layer, it won't inactivate them, it won't kill them. So that's something to bear in mind. We're lucky that we can use alcohol hand gels in this situation, but they don't always work. And other things like bleach and strong UV light are other ways that you can inactivate viruses, kill them.
That's obviously like two things that have come up from across the pond
Yes.
with certain politicians. So he's not entirely wrong, but maybe his means of administering is debatable.
Absolutely.
Right.
It probably would work, but you wouldn't, live to tell the tale.
Yeah, no good killing the virus if it kills you as well. But
absolutely,
it's a solution to killing the virus, right?
Yeah. I got on. I did a radio interview for BBC Radio Humberside a few weeks ago, and a caller had a question about, do they need to wash their clothes at a very hot temperature to inactivate any virus they might have on them? It's actually a really good question. And I said, no, not in this situation. You don't. You can wash your clothes like you normally do, but the important thing is you've got to have detergent in that washing machine like you normally would. You put in a pod or a splash of detergent, and that does pretty much the same thing as these little alcohol gels, and it just dries up those little shells.
Okay, cool.
So, and this is probably my last slide. Virology has absolutely ballooned, not just in the last 4 months, but in the last 20 years or so. And it's now this just umbrella term. And we need, it's high tech, and it's had to get high tech because of these challenges of working with these tiny little things. But we need people who are good at using microscopes and all these fancy microscopes out there, who are chemists that can understand how those little shells form. Who are good at trawling data and writing programs to analyze a huge amount of data we get from viruses. We need epidemiologists. We need people who can predict and react to epidemics and pandemics. And something I left off this list, but something which is really important, are the logistics of dealing with epidemics and pandemics. And it's something which we're having to learn incredibly quickly in the current situation is how do you move reagents around, people around, resources around in an incredibly short period of time.
Yeah, I will just do a quick plug. There is a quantum source coming up on supply chains and pandemic responses.
Oh, excellent. So, I will be signing up.
Watch that space.
It sounds like a huge challenge. Something which often isn't, especially if, from an academic point of view, it's not in the forefront of our minds, but it's probably one of the most important things.
Yeah.
And another thing I didn't mention on this list was people who work with animals and will be able to take samples from animals, in a main way, samples.
Sure.
And identify some of these new emerging viruses that come up. That's really important too. And I've worked with people that have traveled to Africa and actually sampled inside caves and water and things like that, trying to find and predict what the next emerging virus is going to be. One of my colleagues, Shofia Reglui, she was stationed in Sierra Leone during the Ebola virus outbreak. And here's a picture of her and her colleagues trying to very unsuccessfully spell out Ebola. But it was just kind of reiterates how quickly the field has moved from, someone in a lab with ceramic filters trying to purify viruses into this massive multi-skilled approach and to having to respond to these epidemics really quickly.
All right. Well, thanks very much. That was really fascinating, and it's such sort of current insights. It's really cool to have you talk to us. Thanks very much. I don't know whether this is putting you on the spot or not, but what do you think the future of the coronavirus actually is? How are we going to beat it?
Well, it was a pleasure to chat to you today, Adam. I really enjoyed it. It's a tricky one. There are still so many, you wouldn't think so, but there are so many unanswered questions about this coronavirus. The basics. Can it be transmitted any other way than that airborne transmission? What's that R0 number, that reproduction number that you've heard of in the news? The biggie, do people who are infected with this virus develop long-lasting immunity?
Sure.
So I'll probably cop after the bit on your question, but say until we answer some of those basic things, we won't be able to see clearly where we're going in the midterm. I think what we can say for sure is that we are probably going to have social distancing in some form or another for at least another year, maybe even 18 months.
Wow.
And we might get over, flatten that curve, see how it's going, start to relax the measures a bit, and then maybe have to bring them back again. And really all of that is going to be reliant on, are people immune, and the development of a vaccine.
Okay.
Will we be gingerly avoiding big groups for the next while, I think.
Still some way to go then.
Yes. Lots of exciting virology to be done.
Absolutely, yeah. But we'll probably end it there because I'm aware that I've bombarded you with questions throughout your talk anyway. But just to say thanks very much again. And no, it's been really cool and really useful talk.
It's my pleasure. I was happy to do it. Thanks for the invite, Adam.
No problem at all. We'll see you later.
See you later.
Bye. So there we go, some virological insight on how we start beating the coronavirus. I certainly feel much better informed after that chat, and I hope you do as well as being entertained. The best thing we can do is to keep ourselves informed and educated, and if you want more, then Cheryl is willing to take and answer your questions through her Twitter account. You'll be able to find that obviously on Twitter, but we'll also link it through the Quantum Source Twitter feed, Quantum Source Instagram feed, where you'll also be able to find updates on brand new podcasts. And with that, I will thank you for watching. I've been Dr. Adam Booth. You've been great. This has been Quantum Source. Stay well, everyone.